Hello, my name is Neel Kondapalli. I'll be presenting altered temporal and visual resting state network biomarkers in early Parkinson's disease. Parkinson's disease, or PD, is a neurological disorder that affects over 10 million people worldwide. It disrupts motor and non motor driven movement, leading to symptoms including tremor, stiffness, and cognitive dysfunction. The current clinical standard of PD detection relies heavily on observable physical symptoms of the disease, and the implication of this is that the criteria for detection is not as early as it could be. Research into the neuroimaging biomarkers of PD seeks to tackle this very problem by identifying markers of the disease that can be easily and non invasively detected early in its course. Many of the disruptions in higher order cognitive ability caused by PD can be traced to certain dysfunctionalities in regions of the brain responsible for modulating visual and memory related tasks. While studies of the progression of PD have found abnormal activity in these regions, it is unclear how these findings translate to earlier stages of the disease. We theorize that abnormalities in regions associated with the cognitive symptoms of PD may be detectable or at least visible at earlier stages of PD and may be preliminary markers for the development of conditions such as mild cognitive impairment and other cognitive symptoms. Resting state fMRI data for early PD patients and controls was analyzed in this study. The RS-fMRI and T1W anatomical scans were retrieved from the public Parkinson's Progression Markers Initiative, or PPMI, neuroimaging database. RS-fMRI data for 22 baseline visit PD patients and 20 healthy controls was downloaded and preprocessed via the fMRI prep pipeline. An additional custom preprocessing workflow was applied to scrub artifacts and confounding signals. Subject brains were parcellated into 96 different regions. Functional connectivity, or FC, is established when two distinct brain regions are temporally in sync in terms of their neuronal activity. The matrices were thresholded to contain only the strongest 20% of connections. We applied a number of network theoretical metrics to study the structural and functional differences between patient and control brain networks at the global and nodal levels. Modularity and community structure was analyzed on the global level. Nodal metrics included local clustering coefficient, degree centrality, and eigenvector centrality. The network-based statistic, or NBS, was applied to conduct whole brain analysis of functional connectivity differences between the two groups. Persistent homology is a more recent application in FC that does not require a set threshold, and as such is a network modeling approach that analyzes the entire network over multiple threshold values. RIPS filtration is the main driver behind this analysis, and it allows the study of topological invariants such as Betty numbers through multiple network filtration values. The zeroth Betty number represents the number of disconnected components in the network, and the first Betty number represents the number of neural loops in the network. Two simplex participation was also analyzed as an additional nodal metric. All analysis was done using Python. To identify significant differences in functional connectivity between the PD and control groups, the network-based statistic was used. NBS is a non-parametric statistical test used to compare regions between experimental groups across a fixed number of permutations. The FC signatures identified to be significantly abnormal in the PD group were shared by the anterior and posterior divisions of the left superior temporal gyrus, the left occipital pole, the left occipital fusiform gyrus, the inferior division of the left lateral occipital cortex, and the right intracalcarine cortex. These regions demonstrated a significantly different connection strength between the controls and patients, indicating that the presence of PD disrupts normal, healthy functioning in these areas. The connections identified to be abnormal in the patients by NBS may also cause shifts in the general network structure and clustering in patient brain networks. Modularity and Betty curves were used to assess the global network effects on the part of PD. An independent t-test found that modularity between the groups differed significantly. PD patients, on average, had higher modularity than the control group, meaning they had a more segmented network structure. The first Betty curve on the right of the figure shows a rapid increase and spike in the PD group, indicating that the PD networks had more neural loops than the control networks. This suggests that the brain networks of the PD group were more isolated and internally connected than the control networks. Analysis of regional influence through degree centrality and two simplex participation revealed two regions in the occipital lobe, the inferior left lateral occipital cortex and the occipital pole, to exhibit significantly less influence on the global network in PD patient brains. Degree centrality quantifies the sheer influence of a region through the sum of all its connections. And 2, simplex participation quantifies the amount of network triangles a region is a part of. These two regions having a significant decrease in one or more of these metrics means that they played a weaker role in patient brains and may be large contributors to the increased network segmentation detected in prior analysis. The patient brains were observed to be more segmented and globally disconnected. Higher subnetwork segmentation combined with lower regional communication means that the PD patients had more local networks. The inferior division of the left lateral occipital cortex and the left occipital pole were found to exhibit weaker influence in PD patient networks. These two regions are associated with important visual resting state networks. The study found early structural alterations in these networks. Key temporal regions were also identified to have abnormal functional connectivity signatures in the patient brains. The superior temporal gyrus is involved in visual processing tasks and social emotional regulation, and these cognitive processes have been reported to be disrupted in PD prior. The intracalcarine cortex is linked with cognitive functions like spatial reasoning and orienting visual fields, and it is associated with impaired visuospatial ability and has been shown to exhibit altered functional connectivity in resting state PD patients before. In this study, we investigated how network structure in temporal and occipital areas of the brain diverges from normal functioning at an early stage in Parkinson's disease. We identified non-invasive neuroimaging biomarkers in key temporal and occipital regions, along with shifts in global brain network structure that can serve to supplement early detection criteria and advance clinical understanding of the early pathology of Parkinson's disease. Future work involves a longitudinal study to assess how well predicted cognitive symptoms manifest in later disease stages, and analysis of a larger neuroimaging dataset. Thank you.